And welcome dear viewers to the interesting topic on cleaning validation. In this topic particularly we will learn regarding dirty hold time and clean hold time. These are the two major parts and studies required in the cleaning validation. So we will start with the basics and then we will see what is dirty hold time and what is clean hold time and what is the impact of these two hold times in cleaning validation. So as the validation is the establishing a documented evidence for the process, that the process will give the consistent results and will be the reproducible. So similarly cleaning validation is the establishing a written documented evidence that the cleaning method or process or the procedure can and will reduce the residues of the product and the cleaning agents to the acceptable level. That means the cleaning method or procedure, whatever it is adopted, that will clean the equipment properly and it will make the equipment free from the contamination and the residues of the previous product and the residues of the cleaning agents, and it will reduce to a level which is acceptable. So cleaning validation is the validation which supports or which provides the written evidence that the cleaning method is adequate. Then cleaning validation also demonstrates that the routine cleaning and the storage of equipment does not allow the microbial proliferation. That means the microbial growth or increase in the bioburden. So in pharma industry, cleaning is very important. Whenever you are going to a pharma manufacturing plant and whenever you are going to start the batch manufacturing, at that time line clearance will be there, and that line clearance majorly involves the inspection of the equipment and the area, whether the equipments are clean or not and these equipments are proper or not. So from there you can understand that if the equipments are not clean and if the area is not clean, the manufacturing cannot be started. So the people who are working into the pharmaceutical industries, they will understand these things. And the people who are not working into the pharma industry like students, so they should understand that whenever the batch manufacturing is initiated, that time cleaning is checked. Then we will see the importance of cleaning validation. So this cleaning validation gives the validation for the cleaning procedures. That means the cleaning procedures are adequate. These cleaning procedures are effective, and these cleaning procedures and the cleaning method will reduce the residues to the accepted level. So cleaning validation in total is a establishment of the written documented evidence that the cleaning procedures are adequate. So now we will see the hold times. So first is dirty hold time, second is clean hold time, and third one is the sampling delay time. So understand that if you perform a batch manufacturing for tablet formulation, for example, you have taken the line clearance and you have made the batch, manufactured the batch into the granulation area, and now you have lubricated blend in your hand. So now you have used the equipments and your batch manufacturing is over. Now you have transferred the blend to the quarantine area till the next stage, whether it is tablet compression or capsule filling or sachet filling, whatever it is. So now your area where you have manufactured the batch is dirty. Now the next procedure for the dirty area is cleaning procedure. Then the qualified personnel will be there to clean the area. Now suppose they have cleaned the area and the area is ready for the next batch. There may be the batch to batch cleaning or product to product cleaning. This topic I will cover in the next upcoming videos. So for this video you consider the cleaning. Now you have cleaned the area and you got the confirmation from the QA, from the swab analysis or rinse water analysis, that the area is clean. Now that area is clean area and that is on hold till the next batch or next product is manufactured. So that clean equipment or clean area is on hold. On hold means it will be in the clean state till which time. So that is the clean hold time. So now consider the dirty hold time and the clean hold time. Dirty hold time is the time from the end of the manufacturing process until the beginning of the cleaning process. Suppose you have made the batch on the first day of week and on the next day you are going to clean the area. So that area was in dirty state for around 24 hours, for example, or 8 hours or 10 hours or 12 hours. And after cleaning, you have started the next batch in that area after 2 days. So consider here the clean area was on hold for 2 days. That is the clean hold time. So cleaning is done and that clean area was taken into the batch manufacturing, or the batch manufacturing was started in that area for, let's say, after 2 days. So that 2 days will be the clean hold time. So clean hold time is the time from the end of the cleaning until the next use of the equipment or the area. This is also referred as clean equipment hold time. Similarly, dirty hold time or dirty equipment hold time. Now coming to the sampling delay time. Sampling delay time is the time from the end of cleaning until the samples are taken to the analysis. Sometime you have done the cleaning, but after 4 or 5 hours or one shift is there for 8 hours, the samples are not taken. So that sampling delay time will be there for the 8 hours. And why this sampling delay time is required to be considered? Because once you clean the equipment and if you do not sample it, then there are chances of the contamination. So the dirty hold time, clean hold time and sampling delay time, these are very much important factors and parameters in the cleaning validation. So cleaning validation is important from the point of manufacturing, avoiding the contamination, cross contamination, avoiding the microbial growth, and this cleaning validation will give you the documented evidence that whatever the cleaning procedures you have adopted, whatever the clean hold time and dirty hold time you have established, that will not lead to the contamination. And mainly this cleaning validation is very much important and very difficult to achieve in case of the sterile formulations. Dirty hold time is the time from the end of the manufacturing process until the beginning of the cleaning process. That is also known as dirty equipment hold time study, DEST, or simply dirty hold time, that is DST. And this DST, that is dirty hold time establishment, is a part of the cleaning process protocol, that is cleaning process validation protocol. Cleaning of the dirty equipment immediately after the manufacturing process is completed versus the cleaning after DST of, let us say, 24 hours. So what will happen if you leave your equipment in a dirty stage for some days or some hours? What will happen? Sometimes if you leave the area without cleaning for some time, you will have difficulty in the cleaning. Sometimes you will not have difficulty in the cleaning. Sometimes you will feel that if I leave this area dirty for some hours or some time, equipments are getting dried, or the liquid, if the liquid part is involved into the manufacturing, that is getting dried, and the manufacturing uh, and the cleaning procedure is getting easy. But if your product get degraded due to the effect of the light or moisture, then you will have a very difficult time for the cleaning. So DST may or may not have impact on the cleaning effectiveness. Sometimes the cleaning becomes more difficult after some DST, that is dirty hold time, due to the nature of the product. For example, if you are manufacturing a product which get degraded immediately, and if it gives some color after degradation, then you will find that if you leave an area without cleaning, your whole area will be very dirty, very colored area will be there, and you will face problem in the cleaning. Some materials will be hygroscopic, some colored ingredients will be there. Then some materials may also undergo degradation during the whole time due to the light, air and oxidation, and then you will find that the cleaning process is getting very difficult. The DST may impact on the cleaning effective in different ways, like cleaning may become difficult, may become easy, or may not get impacted. So this DST should be specified based on the product nature and the cleaning validation. Then clean hold time. Clean hold time is the time from the end of the cleaning until the next use of the equipment. So if you have cleaned the equipment on 1st January, and if you are going to use the equipment on 1st February, so will it be in a clean state or not? That is the question. Or sometime you may need to re-clean the equipment. So that is the clean equipment hold time, and its study is required. CST is very important as the clean equipment may be affected by contamination, that may be external contamination, or maybe the microbial contamination, that is bioburden. So though it is in clean condition, it may get contaminated. This clean hold time is finalized based on the protocol based studies which are done in the cleaning validation studies, and protocol is there to study the residues after the CST. So once you have cleaned the equipment, that time you will send the sample for analysis, and also after the clean hold time completion, you will send the sample for analysis to study that still the equipment is in the clean condition or not. If the CST is over before the manufacturing process, then cleaning is to be performed. So specific SOPs are there based on the manufacturing area, based on the product, based on the quality systems, total quality management, and the corporate quality assurance. So based on these times are finalized. CST may be given as several weeks based on the validation studies. So how much time you can give for CST? You may give several weeks also. Then coming to the sampling techniques, mainly for the cleaning validation, swab sampling is done. That is known as direct sampling, direct surface sampling, that is swab method. So swab method is there, and rinse sample, that is indirect method. Water is put into the equipment and that is rinsed, and that water is analyzed for the residues. Then what are the best practices? So whenever the equipment is used, the cleaning should be done immediately after completion of the manufacturing activity. Then the DST and CST are required to be established and validated. Then equipment should be cleaned prior to the maximum DST. If you have a dirty hold time of, let's say, 24 hours, so you should clean the equipment before that DST is reached. Then conduct the cleaning validation on the worst case condition. Always have a DST and CST data on the worst case. Then both quality and operations or production requirements are to be considered. See, always there will be a requirement from the production or operation that give the maximum DST so that they can have the flexibility in their operations and production. Then also give the maximum CST. But here the problem of quality will arise. So the CST and DST are to be studied and finalized based on the quality and the production requirements. So this is for the cleaning validation, dirty hold time and the clean hold time. Many of the pharma companies get the queries and the compliance, non-compliance questions due to this cleaning procedure and dirty hold time and the clean hold time. Many of the times regulatory queries are there because of the cleaning, because this cleaning is very important in the pharma field. You might have got good understanding about the cleaning validation and dirty hold time and the clean hold time. So let's meet you soon in the next videos. Thanks for watching the video.